Okay, let's take a look at topic 7, our genetics topic. So let's review about chromosomes. Humans have 46 chromosomes, or 23 homologous pairs. And the term homologous means that each chromosome from one parent has a corresponding chromosome from the opposite sex parent. So if there's 23 pairs, each parent has a chromosome 1, 2, 3, 4, all the way through. So the both of your chromosome number 1, the one you got from mom and the one you got from dad, are similar chromosomes. They're going to have similar genes on them. And they're a homologous pair. They're going to pair up next to each other. Chromosome pairs carry genes for the same traits. Most organisms have 2 genes or alleles for each trait, one from each parent and one on each member of the homologous pair. Sex chromosomes in humans. Females have XX and males are XY. The Y chromosome is much smaller than the X, so it's missing many genes. And this means that many genes on the X chromosome do not have a partner. So that means if a male has a recessive trait on the X chromosome, the Y chromosome will not be able to hide it with a dominant gene. So this makes males more likely to have some traits like color blindness, and these are called sex-linked traits. A common mistake that people make is saying that humans have 23 chromosomes or 46 pairs or some other number combination. You just need to remember it's 23 pairs or 46 total. All right, chromosomes and genes. Each chromosome has hundreds or thousands of genes located on it. Each gene codes for a particular protein. A common mistake that people make is saying genes or DNA are made from protein. Genes carry the instructions to make the protein. The genes themselves are made from nucleic acids. And then a second thing to remember, while genes determine our traits, the environment can have an effect on the expression of our genes. DNA is the chemical that makes up your genes and chromosomes. An analogy for this, it would be if your genes and chromosomes are the instruction manual for your body, the DNA would be the paper that it's printed on. The shape of the DNA molecule is a double helix, which resembles a twisted ladder. And the shape of DNA allows it to replicate or copy itself almost perfectly. DNA, remember, is made of the 4 bases, adenine, thymine, guanine, and cytosine, so A, T, G, and C. And please remember your base pairing rules. A pairs with T, G with C. You can remember apples in the tree, car in the garage, or Akron Tigers, go Cats. And then RNA pairs very similarly, G with C, but then A pairs with U. Adenine will pair with uracil when it's RNA. For protein synthesis, if we need to make proteins from our genes, this is how your genes control your body. So a codon, remember, is a sequence of 3 bases in DNA. Each codon represents a specific amino acid. Ribosomes assemble amino acids in the same order that they are listed on the DNA codons, and then the amino acids will make a protein. The order of the amino acids, which is determined by the DNA sequence, determines the shape of the protein, and the shape of the protein determines its function. Therefore, the sequence of bases in the DNA will determine the function of the proteins in your body. And the proteins are what build and run the body. Okay, looking at messenger RNA, that's going to carry the genetic code to the ribosomes from the nucleus. And the order of the DNA bases in your genes determines the order of amino acids in your proteins, which determines the protein's structure and function. Therefore, how your body functions depends on the order of the bases in your genes. And I know this is repetitive, but it's definitely important to remember this sequence of information. Okay, if we take a look at this picture here, we're going on the left-hand side, starting over here, we have a double-stranded DNA molecule. And it's going to begin to go through replication. So you end up having an enzyme that can pull apart the single strands. And then there are, in the cytoplasm or in the nucleoplasm, you've got extra nucleotides floating around, and they're going to come in and pair up with the available bases. And so you're kind of unzipping the DNA all the way through, breaking these hydrogen bonds in the middle, and then they pair up, and you end up with two identical strands at the end of replication. Down here at the bottom right, we're looking at protein synthesis. So our top picture, you can label that as our double-stranded DNA, and we know this is DNA because it's double-stranded, first of all, but also because it has thymine in there. And then the DNA has to split open, and then messenger RNA can be built off of that DNA. And messenger RNA is only going to be single-stranded. Messenger RNA can actually leave the nucleus and go to a ribosome. And then this bottom section is these are transfer RNA pieces like GGA is transfer RNA, UAC, CUG, and they're going to pair up with the messenger RNA in a specific order. And that's going to tell the ribosomes how to connect these amino acids, and that's going to build that protein. Okay, so now we have mutations. These are changes in DNA. They could be good, they could be bad, they could be neutral. But they can only be passed on to offspring if they occur in reproductive cells, like in a sperm or egg cell. The common mutagenic agents include radiation, chemicals, and viruses. These are the types of things that can change your DNA. A mutagenic agent is anything that can cause mutation. Gene mutations may cause a change in a gene which can change the shape of a protein. And this will have an effect on the way that the protein works, if it even works at all. Gene mutations are caused when DNA bases are in some way changed. You can insert one, you can delete one, you can just substitute one. So it kind of depends on what happens and how it changes the shape. Chromosome mutations are usually caused when a person inherits too many or too few chromosomes. Chromosome mutations affect many genes at once, and most of them are lethal. Down syndrome, which is a non-lethal mutation, is caused by the inheritance of an extra copy of chromosome number 21. And note, it is only chromosome 21 that can cause Down syndrome. It has to do with the specific genes that are located on that chromosome. Okay, moving on to genetic technology. We've got selective breeding, which is the same thing as artificial selection. This is when you're selecting a particular trait and breeding for it. For example, if you want disease resistance or larger fruit or specific colors, you're specifically breeding for that trait. Genetic engineering is when you're making changes to the DNA code of a living organism. You do have to use something called restriction enzymes, which are used to cut apart DNA segments, and then there's another enzyme that helps glue them together. Organisms that receive the new gene will begin to make that particular protein, enzyme, or hormone that's coded for by that gene. The new protein, enzyme, or hormone will be the same as the one that was produced in the original organism. And bacteria are often used because they're simple and they reproduce quickly. The example of gene splicing, or this genetic engineering example that you really should remember, is bacteria that have been engineered to make insulin for diabetics. Remember, we're able to take the human insulin gene and insert it into the bacterial plasmid, and then the bacteria can then make human insulin. Same thing, bacteria have been engineered to make growth hormones for humans, and in both cases the engineered hormones are safe to use because they're identical to the normal human hormones. The picture at the bottom right is an image showing how this process works. So you have to use restriction enzymes to cut out the gene for human insulin, and a restriction enzyme to cut apart the bacterial DNA. And then using something called ligase, you can glue in the human gene into the bacterial DNA, put it into a bacterial cell. That cell can replicate, but at the same time it can produce the protein, insulin, because it has the gene that codes for it. So our new technologies that keep expanding and growing, some of them are called karyotyping, DNA fingerprinting, and they're making it easier to diagnose and treat genetic disease. Although we can't cure all of them yet. So a karyotype is a photograph of an organism's chromosomes. They usually take a picture of their chromosomes as they're going through mitosis or meiosis, and it can determine if a person has a chromosome disorder like Down syndrome. In this diagram here, you can see that this person has 23 sets of chromosomes, and they do all appear to be normal. This person is a male because they have an X and a Y chromosome. But the rest of their chromosomes, even though they're kind of bent in shape, that's just how they looked in the cell, but they should all be similar in size and shape. There's no extras or none missing. So this person has a normal karyotype. DNA fingerprinting, or also called gel electrophoresis, will create banded patterns based on a person's DNA sequence. Each fingerprint is unique, so it can be used to identify people. Fingerprints of relatives are similar to each other, so it can be used to determine genetic relationships between two people or even two groups of organisms. Genetic research has posed many ethical problems, the right and wrong of certain things. And that's something that science alone can't answer. So the definition of ethics is the study of moral standards and how they affect our conduct or our behavior. That takes us to the end of our topic 7. You can go ahead and complete your homework for this topic.